The bone sits in a sterile tray under fluorescent light in Leipzig, Germany. It is smaller than a human pinky. A technician drills into it, extracting powder, milligram by milligram. Less than 10 milligrams total. The machine hums. Sequences begin assembling on a screen, letter by letter, hundreds of millions of them. And the geneticist reading the output stops, because the code on this screen does not match any human being alive today. It does not match any human being we have ever cataloged. Not modern, not Neanderthal, something else entirely. That bone fragment came from a cave in the Altai Mountains of southern Siberia, a region so remote it takes a 10-hour van ride from the nearest major airport to reach it. And what it contained would force us to redraw the human family tree, not with fossils and skulls and careful morphological comparison, the way we have always done it, but with nothing more than a whisper of genetic code trapped inside a sliver of bone the size of a pencil eraser. I want to tell you how that happened. Because the story of the Denisovans is not just the story of a discovery. It is the story of how the tools we use to ask questions determine the answers we are able to find. And for most of the history of paleoanthropology, we were asking with the wrong tools. For the better part of a century, identifying a new human species required bones. Skulls, ideally. Jawbones, femurs, pelvises. You needed something a trained anatomist could hold in both hands, measure with calipers, compare point by point to known specimens, and place on a branching tree of human evolution. This is how Eugene Dubois identified Homo erectus from a skullcap and femur in Java in 1891. How decades of Neanderthal finds across Europe and the Middle East confirmed a distinct lineage. How Lucy's 3.2-million-year-old skeleton changed our understanding of bipedalism. The method worked because the question was physical. Does this skeleton look different enough from everything else to warrant its own category? If yes, new species. If no, variant. And by the early 2000s, the consensus tree felt reasonably settled. Modern humans, Homo sapiens, emerged in Africa roughly 300,000 years ago. Neanderthals occupied Europe and Western Asia until about 40,000 years ago. A few enigmatic fossils from Asia, Homo erectus surviving late in Indonesia, the tiny Homo floresiensis discovered on Flores in 2003, filled out the eastern edges. The basic architecture seemed clear. 3, maybe 4 major lineages outside Africa in the late Pleistocene. The branching pattern was understood. The cast of characters was known, everything accounted for. But that confidence rested on an assumption so fundamental that nobody thought to question it. The assumption was this: if a human species existed in significant numbers across a significant range for a significant time, we would find their bones. Think about what that requires. It requires that bodies be deposited in environments conducive to fossilization, that those environments survive geological upheaval, that someone eventually digs in the right place, that the bones, once found, are recognizable as something new. Every link in that chain can break. And for one entire branch of the human family, every single link did break, for a very simple reason. Denisova Cave sits at the junction of Siberia, Kazakhstan, Mongolia, and China, overlooking the Anhui River. It is not particularly large, maybe the footprint of a modest apartment. But it has high ceilings, a natural limestone chimney that provides ventilation, and year-round cool temperatures that slow biological decay. For at least 200,000 years, it was an attractive shelter. Russian scientists discovered archaeological remains there in the 1970s and have been excavating ever since. 22 distinct sediment layers, stone tools spanning multiple traditions, Mousterian scrapers associated with Neanderthals, Levallois points requiring considerable planning and skill, even a bone-sewing needle estimated at 50,000 years old, 7 centimeters long, drilled and polished from bird bone, personal ornaments made from polished dark green chlorite, mammoth tusk, and animal teeth. Whoever lived here was not primitive. The artifacts made that clear. But the bones. The bones were a problem. The cave had been home not just to humans, but to hyenas, wolves, and other carnivores that cycled through over tens of thousands of years. They chewed, crushed, and scattered skeletal remains into thousands of tiny, unidentifiable fragments littering the cave floor. A micro-CT scan of one specimen later showed acid etching and pitting on its surface, evidence of having passed through a hyena's digestive system. You could hold one of these fragments in your hand and have absolutely no idea whether it came from a mammoth, a deer, a sheep, or a person. The morphological method, the one that built the entire field of paleoanthropology, was blind here. And so, for decades, those fragments sat in storage boxes, essentially invisible to science. In 2008, during excavations in Layer 11 of the East Chamber, Anatoly Derevianko and Michael Shunkov of the Russian Academy of Sciences found a small fragment of a finger bone, a child's finger. The growth plate was not fused, suggesting the individual was between 5 and 7 years old. The bone was catalogued, placed in a box marked Modern Human, and shipped to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. Svante Pääbo's lab had been working on the Neanderthal genome. They were screening bones from multiple sites. This one was not expected to be special. Actually, let me back up for a moment because this part matters. The reason Pääbo's lab could even attempt what came next is the result of almost 30 years of methodological work. Ancient DNA degrades. It breaks into tiny fragments. It gets contaminated by bacteria, by the DNA of anyone who has ever handled the bone, by microorganisms in the soil. For most of the 20th century, extracting usable genetic material from anything older than a few thousand years was considered impossible. Pääbo had been chipping away at that barrier since the 1980s, starting with Egyptian mummies, developing cleanroom protocols comparable to the computer chip industry, creating computational tools to separate ancient sequences from contamination noise. By 2010, his team had just published the draft Neanderthal genome. They had the infrastructure, the expertise, the pipeline. So when the finger bone from Denisova arrived, they did what they always did. They drilled into it, ground the sample to powder, applied chemical baths to strip away millennia of contamination, and extracted mitochondrial DNA, the smaller, more abundant genetic material inherited through the maternal line. The results, published in early 2010, were baffling. The mitochondrial sequence diverged from modern humans by roughly twice as many substitutions as the Neanderthal sequence did. It was not modern. It was not Neanderthal. It sat on its own branch entirely. The team initially called the specimen X-woman, and I remember when I first read that paper, I assumed it was going to turn out to be some kind of contamination artifact or a deeply divergent Neanderthal. It did the opposite. David Reich at Harvard, Richard Green at University of California, Santa Cruz, and the rest of Pääbo's international team moved rapidly to nuclear DNA. The nuclear genome, sequenced to 1.9-fold coverage from less than 10 milligrams of bone, confirmed it. This was a distinct lineage, a sister group to the Neanderthals that had split off more than 390,000 years ago, a whole branch of the human family tree hiding in plain sight. They named them Denisovans after the cave. And then they found something even stranger. When the team compared the Denisovan genome to populations around the world, they expected to find traces of interbreeding with nearby Asian populations. Instead, the strongest signal appeared in Melanesians, people living in Papua New Guinea and nearby islands, thousands of kilometers to the south. 4 to 6% of their genome was Denisovan, not Neanderthal. Denisovan. This meant the Denisovans had not been a small, isolated population in one Siberian cave. Their range had to have extended across a vast stretch of Asia. From Siberia to Southeast Asia, far enough to encounter the ancestors of modern Melanesians as they migrated through. And if you have been following this channel for a while, you know what this means for the fossil record. It means an entire human species, widespread across the largest continent on Earth for tens of thousands of years, left almost no recognizable skeletal trace. A few teeth, a finger bone, fragments chewed beyond recognition by cave hyenas. That is it. The bones existed. We just could not see them. This is where the story could have stalled. A genome, some teeth, and a profound gap in the physical evidence. But the tools kept evolving, and one of them, I think, deserves more attention than it gets. In 2012, Samantha Brown, then a graduate student at Oxford, working with geochronologist Tom Hyam, began applying a technique called ZooMS, zoo archaeology by mass spectrometry, to the thousands of unidentifiable bone fragments from Denisova Cave. The method works by extracting collagen proteins from bone, breaking them into peptides, and reading their mass signatures. Different species produce different collagen fingerprints. You cannot tell a human fragment from a sheep fragment by looking at it. But you can tell them apart by weighing the molecules inside. Brown and her team ran over 2,300 fragments through this pipeline. The vast majority were animal, but a handful came back human. And one of them, catalogued as DC-1227, a sliver of long bone weighing 1.68 grams and measuring just 24.7 millimeters at its longest, changed the story again. If you are finding this useful, if this kind of deep look at the science behind the headlines is something you want more of, subscribing is the single best way to make sure we can keep doing it. We are working on a follow-up that goes deeper into how ancient interbreeding shaped the modern immune system, and it connects directly to what we are covering today. Back to DC-1227. Mitochondrial DNA analysis at the Max Planck Institute showed Neanderthal ancestry. But when Viviane Sloan sequenced the nuclear genome, she found something she could not explain. The nuclear DNA was split, roughly equally, between Denisovan and Neanderthal sequences. She thought she had made an error. She repeated the extraction 6 times. Same result every time. In 2018, the team published their conclusion in Nature. DC-1227, now known as Denisova 11, nicknamed Denny, was a girl who had lived approximately 90,000 years ago. She was at least 13 years old when she died. Her mother was a Neanderthal. Her father was a Denisovan. She was a first-generation hybrid, the direct offspring of 2 entirely different human species. Let that sit for a moment. Among the tiny number of archaic human specimens ever sequenced, one of them turned out to be the child of parents from 2 different species. Pontus Skoglund, a population geneticist now at the Francis Crick Institute in London, called it a landmark find, and the implications cut deep. First, what the evidence shows. Denny's genome is not a statistical abstraction or an inference drawn from population-level data. It is a direct record. 50% Neanderthal chromosomes, 50% Denisovan. Her Denisovan father also carried traces of Neanderthal ancestry in his own genome, meaning interbreeding had happened before in previous generations. Her Neanderthal mother belonged to a population more closely related to later European Neanderthals than to the local Altai Neanderthals, suggesting westward migrations into Siberia. Second, what it rules out. The old assumption was that interbreeding between archaic human groups was vanishingly rare, an anomaly, a statistical ghost in population-level data. Finding a first-generation hybrid among such a small sample of sequenced genomes makes that assumption nearly impossible to sustain. We have sequenced nuclear DNA from fewer than 24 archaic human specimens total. The odds of stumbling onto a direct hybrid by chance, if hybridization were truly rare, are astronomically low. As anthropologist John Hawks at the University of Wisconsin-Madison put it, hybrids must have been far more common than anyone assumed. They must have been, in his words, nowhere near as rare as people had been thinking. Third, what it implies. These were not isolated populations bumping into each other once in 100,000 years. Neanderthals and Denisovans, and eventually modern humans, were meeting, mating, and producing viable offspring with some regularity. The boundary between species was more permeable than the textbooks suggested. Human evolution was not a clean branching tree, it was a braided river. I rewrote this next section 3 times because the evidence genuinely pulls in 2 directions, and I want to be honest about that. On one hand, the genetic data clearly shows interbreeding happened. On the other, the Denisovan and Neanderthal genomes remain distinct enough that we can tell them apart with confidence, which means the groups did not merge into one population. They mixed at the edges. They remained mostly separate. The debate now is about frequency, about geography, and about what drove those encounters. Some researchers suggest Denisova Cave itself was a borderland, sitting at the far eastern edge of Neanderthal range and the far western edge of Denisovan territory. A place where both groups occasionally converged, a kind of prehistoric crossroads. Others argue the mixing was more widespread, but that we simply lack the ancient DNA from enough sites to see the full pattern. We have high-quality genomes from exactly one cave. That is an extraordinarily small window into tens of thousands of years across millions of square kilometers. And there are bigger unknowns that I find genuinely fascinating. We still do not know, with confidence, what the Denisovans looked like. For over a decade after their identification, we had almost no physical remains large enough to study morphologically, just teeth and bone splinters. A few researchers attempted to predict their appearance using DNA methylation patterns, comparing gene activity signals to known skeletal effects in modern humans. The results suggested a wide skull, a protruding jaw, and a broad dental arch. But predictions from methylation data are rough. They give you a silhouette, not a portrait. I think this might be the most underappreciated oddity of the whole story. We sequenced an entire genome, mapped their genetic legacy in billions of living people, traced their range across a continent, and for years we could not draw their face. But then in 2014, geneticist Rasmus Nielsen and his postdoc Emilia Huerta Sanchez at UC Berkeley found something that deepened the mystery further. They sequenced a gene called EPIS1 in 40 Tibetan and 40 Han Chinese individuals. EPIS1 regulates the body's response to low oxygen, specifically by controlling how many red blood cells the body produces when oxygen is scarce. It is the single strongest signal of natural selection ever documented in a modern human population. 87% of Tibetans carry the adaptive variant. Only 9% of Han Chinese do. It allows Tibetans to live permanently above 4,000 meters, where the air carries 40% less oxygen than at sea level, without the dangerous overproduction of red blood cells that would thicken the blood and cause stroke, pulmonary edema, and pregnancy complications in most other humans. When Nielsen compared the Tibetan version of EPAS-1 to genomes from populations around the world, including every population in the 1,000 Genomes Project, he found no match. Not in Europeans, not in Africans, not in other Asians, not in Melanesians, despite their high Denisovan ancestry. Only one genome in the entire database carried the same haplotype, the Denisovan genome, extracted from a finger bone in a Siberian cave. The match was too close and too long to be explained by shared ancient ancestry. It had to have been introduced by interbreeding within the last 40 to 50,000 years. Somehow, an encounter between modern humans and Denisovans, likely in Central or Eastern Asia before Tibetans and Han Chinese diverged as populations, had passed along a gene that now allows millions of people to survive on the roof of the world. A genetic gift from a species we never even knew existed until 2010. And here is a side note, but it connects. In 2020, Pabo and Hugo Zeeberg found that a major genetic risk factor for severe COVID-19, carried by about half of people in South Asia and 1 in 6 Europeans, was inherited from Neanderthals. Ancient interbreeding is not just a curiosity of the deep past. It is shaping modern medicine. And as of 2025, we may finally be putting a face to the name. A massive, well-preserved skull found decades ago near Harbin, in northeastern China, and hidden at the bottom of a well during the Japanese occupation to protect it, has been tentatively linked to the Denisovans. Researchers initially classified it as a new species, Homo longi, dragon man, after the Black Dragon River province where it was found. The skull is striking. A brain case of over 1,300 cubic centimeters, larger than the modern human average. Massively developed brow ridges, wide eye sockets, a large mouth, no chin. It was estimated at 146,000 years old. When Chao Mei Fu, a paleogeneticist who had been part of the original Denisovan discovery team years earlier, extracted proteins and mitochondrial DNA from dental calculus trapped on the skull's teeth, the genetic signal pointed toward the oldest Denisovan inhabitants of the Siberian cave. 3 protein variants matched sequences unique to Denisovans, and in September 2025, remains from 6 additional sites in China were proposed to belong to the same species. If confirmed, and the work is still ongoing, this is the moment the Denisovans finally step out of the data and into the physical world. A species discovered through a machine reading code from a finger bone the size of a pencil eraser, now staring back at us from a skull that spent decades hidden at the bottom of a well in northeastern China. That skull is being studied right now. More protein analyses are underway. New techniques for recovering nuclear DNA are being developed. Somewhere in a lab, a sequence is assembling on a screen, and someone is reading it for the first time. And we are about to learn whether Dragon Man and the girl from the cave share the same bloodline. The Denisovans hid from us for 250,000 years, not because they were rare, because we were looking with the wrong eyes.